Hello, folks. Today we are going to talk about cellular respiration, the entire process of how we take the energy out of glucose, which is C6H12O6, and turn it into the energy of ATP. And of course, we don't just get like one ATP out of glucose. Our goal is to get as many as possible out of the process, and it turns out we can get probably between 36 to 38 ATPs out of each molecule of glucose, which runs a lot of things. So this is a very important process, but to do so, we are dealing with our laws of thermodynamics because we're trying to convert energy while minimizing our second law of thermodynamics, the loss of said energy from one place to another. So we have to deal with activation energies, we have to deal with different types of energies. And so as we go through these processes, keep an eye on a couple of things. One, keep an eye on every energy conversion we make and what type it is. So we'll have energy conversions that are purely chemical movement, which typically are the movement of a phosphate group because it's a high energy group. We'll have energy conversions that are redox, so that's our electron movement from one place to another. We'll have energy movements that are positional and gradient based. So those are all things. And then, of course, we also want to watch for our energy and our enzymes. So we're going to watch for each enzyme and how it works in many of these cases. And then finally, the other thing we want to watch for is what's happening to our carbon in the process. All right, so we'll start with the process of glycolysis. So here's the entire process of glycolysis. I know it's a lot of pieces, but we're going to break it down and play around with it a little bit, and that should help. So this is the process of glycolysis. Here's our glucose molecule. Starts right over here, our C6H12O6, and you can see it's in essentially what would be a standard conformation for glucose, which is the hexagonal boat shape. So although we don't get too much into the actual structures in a lot of the early classes, it's pretty important, and it's a pretty stable shape. Glucose is a fairly stable molecule. To get it to actually break down and do things takes a fair amount of work to extract the energy out of those bonds. And in fact, you'll notice the very first thing that happens is an energy exchange in the direction of taking energy that already exists. So in this case, we're going to turn an ATP into an ADP and attach a phosphate group to the glucose. This is done using a hexokinase. So kinase for moving a phosphate group, hexo because it goes on the 6th carbon. So essentially, this enzyme allows our carbon chain to bust right over here, and it tacks a phosphate group onto it, bringing it off the ATP. So the energy from breaking the ATP goes into attaching this phosphate group. The next thing that happens, phosphoglucose isomerase, means that we're taking a phosphoglucose, this molecule, and making it into an isomer. So you might notice that there's not a lot of difference between these two molecules, but up here, there's a double bondo, and now we've moved it to here. Now that's key because a double bonded oxygen is actually a fairly strong bond. And we really want to be able to use that outside end to create something that's easier to work with. Because as you can see, the next move is to do the same thing we did a moment ago, but on the other side. Phosphofructo. So now it's a fructose 6-phosphate. Fructose and glucose are almost the same, except for how they're put together in terms of where the oxygens attach. Kinase, so we're taking our ATP, turning it into ADP. So you've noticed we've used some energy in this process and attached a phosphate group on the end. So now our long carbon chain has 2 phosphate groups, one on each end. Now, phosphate groups are very reactive. So when they're on the ends of a molecule, they're really going to pull on that molecule. They are like all the electronegativity. It's pretty much some kind of complex tug of war. And so with all of that happening, it weakens the bond in the middle. And it takes just one enzyme to then be able to break it apart. In this case, it's fructose bisphosphate aldose, whose whole job is to take fructose and chop it in half. Kaboom! Now we have 2 pieces. Now sometimes it's a little hard to follow these 2 pieces, but this is the top piece, this is the bottom piece, and they're 2 slightly different molecules, even though they contain pretty much the same pieces. This one is dihydroxyacetone phosphate. This one is glyceraldehyde 3-phosphate. And you'll notice the only difference between them is, again, where that double bond oxygen is. And there's an enzyme that just actually converts them between the two, which is good because we only need one of them. That's another isomerase. So, making an isomer. So at this point, we have put energy into this equation by 2 ATPs, and we've taken a 6-carbon hexagon and broken it into 2 3-carbon straight chains. So we've used energy, but we've made smaller pieces. That's important. All right, so what's the next thing we're going to do? Well, we're going to take this glyceraldehyde 3-phosphate, and we're going to hit it with a dehydrogenase. We're going to take off a hydrogen. What we're going to do with that hydrogen, it turns out we're going to stack it onto this thing, Na-D-nicotinamide. You know what? I'll let you look it up later because I'm not going to say it correctly. NAD is honestly what we call it, and in this case, NAD+. And then when we add the H to it, we call it NADH. So, what do we see here? Well, if it's a dehydrogenase, it took away a hydrogen. Hey, wait, isn't taking away a hydrogen an oxidation reaction? So we oxidized this molecule. That's important. If you oxidize, you must reduce. We reduced the NAD plus to NADH. We both made the charge more negative and gave it a hydrogen. So that's definitely reduction. So this is our electrical energy. So this enzyme allows us to store energy in the form of NADH. Okay, so now that we've done that, by the way, we do that twice. That's really important because, don't forget, we have 2 of these little 3-carbon pieces, and so this one becomes that one and does the same thing. So we're going to go through the second half of this whole thing with 2 molecules, so everything doubles up. So we have 2 NADHs. But let's look at the process first. So now what we have is what's called 1,3-bisphosphoglycerate. If you're trying to take that apart, glycerate essentially means a 3-carbon chain, each attached to an oxygen. Bisphosphate means 2 phosphates, and 1,3 means one of them is on the 1 carbon and one of them is on the 3 carbon. So the words are always big, but it's not that hard to take apart. Hey, before when we had phosphates, it made everything a little more reactive. We used it to break apart a molecule. In this case, we're not going to use it to break apart a molecule, but we are going to use it to store some of this energy. So our redox reaction gave us the energy to put this phosphate group on the carbon chain. The energy of being able to break that phosphate group off the carbon chain allows us to store an ATP. And here's another kinase. You'll notice there's a kinase every time we deal with an ATP. There's 2 of them up here, 2 of them down here. And so this time, though, we stored an ATP. That's exciting. It leaves us with a molecule that is now only has 1 phospho, so phosphoglycerate. We mutase it, which in this instance is essentially another, similar to an isomerase. So it's just changing the location, and in this case, we're putting the phosphate group in the middle. We're trying to make this phosphate group want to fall off, really. That's really a big part of what we're trying to do, because, well, if it's easy for it to come off, we can store it as ATP. That's part of the idea, is to capture this energy as ATP, so we're making this molecule more energetic, grabbing hold of something, and storing it away. In between, we have 1 enolase. So enol is often how we end, for example, alcohols and things like that. So OH, only in this case H2O, so it takes an entire water molecule off. So you'll see we've removed a water molecule. It's from down here. There we go. And it's over here now. So we take that away. We end up with phosphoenol pyruvate. So there's the phospho group. And the pyruvate is essentially the 3 carbons attached to the double bond OH. So you might also see it if it has the H attached as pyruvic acid, because it's got a carboxyl or carboxylic acid group on the end. But it's got a phospho, so phosphoenol pyruvate. And again, we're going to break off this phosphate group, store it on an ATP, one more kinase, in this case pyruvate kinase, because we create pyruvate at the end. So what did we do here? Well, we took our original phosphate groups and we were actually able to store 2 of them onto ATPs. But wait, because we had 2 3-carbon molecules, we did this twice. So in fact, we stored 4 phosphate groups onto ATPs. That's pretty exciting. Mostly it's exciting because way up here, we had to give up 2 phosphate groups just to give the energy to get this started. So if we gave up 2, we'd want to make back more than we gave up to make this process worthwhile, and we do. We make back 4. So net production is 2 ATPs from 1 glucose molecule, and we are left with 3 carbons in an organic form that honestly is not only not that useful to us, but can get toxic if we're not careful with it. So, while glycolysis is an okay process, it's definitely not the end point that we are looking for. Other thing to watch from here is that this NADH is still hanging around. Keep an eye on that. We may need it for later. All right, so what's going to happen to that pyruvate now? Because like I said, we don't actually want it to do a whole lot of just hanging out. If it hangs out in the body, it actually gets kind of turned around in different ways. It can turn into lactate, which can turn into lactic acid, which can cause us all kinds of problems. We don't actually want to keep organic acids around the body. Got to do something with it. Here's what's next. All right. So now we have pyruvate oxidation. So pyruvate oxidation is pretty much exactly what it sounds like, which is we're going to take a pyruvate molecule, so you might remember that's what was left a moment ago, and we're going to oxidize it. I wonder what exactly we're going to do by that. Well, to oxidize, it means we either need to add oxygens or remove hydrogens or move some electrons around, and we are indeed going to do that. I believe the enzyme here, which I don't have listed on this, is pyruvate, another dehydrogenase enzyme, either that or it's a decarboxylase enzyme, because what this does is it actually removes a carboxyl group. And in this case, a carboxyl group is CO2, carbon dioxide. Hey, I wonder what happens to CO2 in the body. Well, it's a gas. We know gases can diffuse through cell membranes. So no matter where this happens, if there's a CO2 floating around, it actually can just kind of float out. And in most of these cases, because of the sort of pressure of gases, it'll float into the bloodstream, and those little blood cells are actually made to carry CO2. It dissolves well in liquids, so it'll find its way out, mostly back to your lungs. But the cell doesn't worry about it because it does that on its own. You'll notice we do another thing here. This NAD plus becomes another NADH. See, that's why I think it's a dehydrogenase, because I'm pretty sure we took a hydrogen molecule and added it to this NAD. So we took off a CO2, we added a hydrogen to NAD, and now our end product is still not quite what we want it to be, but we're getting closer. So we have an acetyl-CoA at the end. Acetyl has 2 carbons. We started with 3 carbons of pyruvate. We end with 2 carbons of acetyl. It has only 1 oxygen for those 2 carbons. So this one over here had 3 for those 2 carbons. So we actually have managed to cut back the pyruvate because we oxidized 1 carbon and made it go away. The last thing you'll notice we did is we attached it to something, in this case a coenzyme A group, which attaches via a sulfur. The sulfur is not that important, it's just how it attaches. But the coenzyme is, because the coenzyme A allows it to get to where it's going next and puts it in the right spot for the enzymes to grab it. So we get an acetyl-CoA. Acetyl on its own, not overly useful. In fact, it's the base of acetic acid if it picked up another oxygen, so it could be problematic. But attached to the CoA, now we can do something with it. All right, so in terms of energetics of pyruvate oxidation, we do have an oxidation reduction reaction, and we do have a reduced molecule that we haven't done anything with. So we have something energetic happening. But mostly we haven't dealt with anything ATP related in this case. So we're still at only a net of 2 ATPs. Now let's take a look at where this acetyl-CoA is going to go. This lovely insanity of many, many different carbon compounds is, technically it has 3 names. My preferable name tends to be the citric acid cycle. So I will almost always call it the citric acid cycle because it starts with citrate, which is citric acid. It is sometimes also called the Krebs cycle because Krebs is the guy who discovered it. And then sometimes you will see it called the tricarboxylic acid cycle. Or sometimes tricarboxylic acid is cut back to TCA. That is another fairly common one nowadays. A lot of people are using TCA cycle for what this is, but it's all the same. And the reason they call it that is tricarboxylic acid because there are carboxylic acids that get butt off of this as we go. All right, let's go back to our acetyl molecule. Where'd it go? It's way up here at the top. This is our C2. So don't forget we have 2 carbons left from our pyruvate. Now, as a reminder, we had 2 pyruvates. We have 2 acetyl-CoAs. We're going to go through this cycle twice. That's a key. But the first thing we do is that coenzyme allows it to come over to the cycle, and it helps to attach it to citrate. Well, actually, it helps to attach it to oxaloacetate, which is the previous one. This one has 4 carbons. 4 carbons plus 2 carbons. That is how we get our 6-carbon citrate molecule. So here is our 6-carbon citrate molecule. You'll notice it has a lot of COO minuses around it. So those are those carboxyl groups, carboxyl, 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 tricarboxylic acid. And, well, we're going to try to get rid of a few of those. All right. I don't have all of the enzyme names for this one because this one can get a little weighty, but we can still follow the process pretty easily. So, for example, the first thing that happens is citrate goes to isocitrate. Clearly, we just need to make an isomer. We're trying to make something typically more reactive or easier to react. So what have we done here? We took this OH off the middle carbon and popped it down on this bottom one. So now we have an O here, a carboxylic acid group here. This carbon has electronegativity pulling all over it. It is going to be much easier to pop one of those bonds with not as much energy, and therefore that energy can more easily be captured. And in fact, that's what's going to happen here. So you'll notice from C6 to C5 does involve the loss of one of the carboxylic groups. In fact, it's this one in the middle. See, the 2 on the ends are still there. So we have a CO2 that's going to float away just like the last one. So where did the energy come from to do this? Well, I see another redox reaction. We moved some electrons around, captured another hydrogen. So this is a reduction. So we oxidized the carbon, thus getting the CO2. We reduced the NAD, thus getting the NADH. So that was our energetic change here. Now we have alpha ketoglutarate, which is a really neat molecule. It has 5 carbons, and we're going to do the same thing again. We're going to do another oxidation reduction reaction. We are going to take off one of the groups. In this case, I'm fairly certain it's the bottom one that gets pulled because you can still see the top one here. And the energy of breaking that bond and getting that carbon out is captured in this NADH. So the carbon is oxidized, the NAD is reduced, and the reduced molecules are holding the energy. So what's happening? We're holding the energy by moving the electrons. This makes succinyl-CoA. So at this time we attached another coenzyme A. Coenzyme A is a pretty important coenzyme. You need it. To get from succinyl-CoA to succinate, it takes off the coenzyme A, and actually it uses the energy of breaking that coenzyme off to grab a phosphate group and attach it to something. So you'll notice these are a little different. GDP, GTP. It's not quite ADP, ATP. This is guanine instead of adenosine, but it's still an energetic molecule. And actually, there are some microbes that really can fully transfer this to energy, just like ATP. In our case, I usually say think of it, but don't necessarily count it. Plus, you're only going to get 2 of them, so it's not that important. All right. So now we have succinate, which is a C4 molecule. We are going to do another redox reaction. So we're trying to capture a little bit more energy out of some of these bonds. In this case, we're going to break off some hydrogens. In breaking them off, we create a double bond with the carbon, and we actually grab a slightly different molecule here. We turn FAD into FADH2. So that's more reduction, oxidation. This is now oxidized. That makes fumarate. And by the way, you'll notice that a big part of what we're trying to do now is kind of put things back where they belong, but again, without losing any of the energy that was involved in tacking those 2 carbons on there. We want to milk every bit of energy from those original 2 carbons as we can. And so to do that, there's these very small changes, each of which allows for either minimal energy loss or some energy capture. So here we captured a little energy. This is the fumarate. And then the fumarate, which has our double bond, goes to the malate, which actually breaks the double bond. It adds water. So this would be a hydrolysis of the double bond. So that is another energetic type of reaction. And in there, we're going to capture one more piece of energy before going from the malate to the oxaloacetate by turning that into a double bond. And in creating that double bond and breaking off that hydrogen, we're able to capture that last hydrogen and tack it on an NAD. Then we're back to oxaloacetate, and it can grab the next acetyl-CoA and keep going. So the bonus to the loop is that we don't lose anything in it except for the thing we added. So we add 2 carbons, we get 2 carbons out as CO2. By the way, once you run this loop twice, we lost 2 CO2s for each of our pyruvates a moment ago. We lose 2 for each run of this circle. 1, 2, 3, 4, 5, 6. That's all 6 carbons of our glucose are now off as CO2. So ignoring all the rest of these carbons, we have now fully oxidized our glucose. And in doing so, we also reduced a whole bunch of random molecules. They seem random. They're coming back, I promise. So we have a whole bunch of NADHs, NADH, NADH, FADH2. In fact, don't forget 2 runs. So we're talking about 6 NADHs and 2 NADH2s. That's a lot of reduction to deal with. And maybe even a little bit of energy over here. So this is a pretty good process because it creates a lot of energetic molecules, but they're not really currently able to be used for energy. Breaking the NADH is not sort of an easy thing to do. It doesn't give us the kind of energy ATP does. So we still want to turn this all into ATP. So what's next? We no longer have a glucose. We've fully oxidized it to CO2. We do have a bunch of reduced molecules, and we only still have a net 2 ATP. Time to really kick up our game and do something exciting. And that's why it's time for the 4th major step in the process of cellular respiration, the electron transport chain. So we hadn't talked too much about where these things are happening. Glycolysis can happen anywhere in the cytoplasm of a cell. Everything else happens in the mitochondria. And so most of the time, these molecules, especially the pyruvate, moves into the mitochondrial matrix, and therefore it is able to be oxidized in the mitochondrial matrix. The citric acid cycle happens in the mitochondrial matrix. That's the very, very inside of the mitochondria. So you might recall that a mitochondria, usually we kind of sketch it like this, and then it has a membrane inside of it. Whoop. The matrix is all the way inside. So the matrix is all the way in here. This is the inner membrane, And this is the outer membrane. And this is the intermembrane space. And that's where we're looking at here. So the mitochondrial matrix is where the citric acid cycle happens. So it's where all of our NADH and FADHs are. It's going to interact now with the actual membrane. And it's going to push some things across to the intermembrane space. And again, we don't care about our CO2s, even if they're formed in here, because they are able to diffuse through these membranes and they will find their way out of the cells and back into the bloodstream. Or honestly, some of them probably just literally drift out of your body. Did you know that? If you pump CO2 in your body, it'll just diffuse out. It just might take a while. That's a thing that happens sometimes with the laparoscopic procedures. All right, but let's look at this electron transport chain because, well, we have all this captured energy in the form of reduced molecules, but what do we do with that? What does it mean? Like, what on earth? What have we done? We've essentially made little batteries. So we've shoved the energy into the little batteries, but I mean, holding a battery doesn't give you energy. You can't just go like this to a battery and say, oh, well, I got energy now. You have to actually attach it to something that can extract that energy from it. That's this process. The electron transport chain is going to extract the energy from the reduced molecules, and it does it in this really interesting way. You ready? Okay. So, as we've mentioned before, when we say reduced and oxidized, sometimes we're talking about hydrogens and oxygens, but we're also talking about electrons. And a reduced molecule, grr, Leo the lion says grr, has gained electrons. So what we want to do is we want to use that electron through a circuit to provide us with energy. And that's what happens here. This NADH has an electron that it is going to release to this protein complex. And without getting too much into the details of how it bounces around, it pretty much hits the edges of atoms and then goes to other atoms. That electron is able to move to this complex. It's able to move to the number 3 complex. It moves through a cytochrome, and cytochromes are neat because they have heme groups, so ions, just like our bloodstream does. Again, places where electrons can jump. It goes through the number 4 protein, and then, as you know, a circuit really needs to be complete. You can't just have electrons floating all over the place. They're going to bombard something and break it into pieces. Electrons are problematic. That's why you don't stick your fingers in sockets. So this electrical energy needs to land somewhere. Where does it land? It lands on oxygen. Oh, wait, we've been talking about cellular respiration this whole time, and we had not actually talked about oxygen gas. We had oxygens as part of our original glucose. They are what went off in our CO2s. We added some water and things to them. We hadn't actually dealt with oxygen gas, but here the oxygen gas is what we call the electron acceptor. It picks up those electrons. It picks up a couple of extra hydrogens. It turns it into water. That is a safe way to ground the electrons. So you end up with water. You don't end up with electrons floating around. Okay, so what was the purpose of that? Why are we creating a little wire of electrons? Well, just like when you add a battery to something and the energy goes through the wire to do something, we want to accomplish work. We need to use the energy of this electron moving, so that's our kinetic and electrical energy there, to, in this case, push something else. Remember those hydrogens? Here's our goal. We're going to push hydrogens through these proteins. So these proteins are actually channels. Most of them. 1, 3, and 4 are channels. 2 is picking up the electrons from FAD, and the cytochrome C is just an electron shuttle. But the other ones are channels, and they are pushing hydrogens through them. Every time the electron goes through, it also pushes a hydrogen ion through. So what's really happening up in the intermembrane space is that we're collecting a whole bunch of hydrogen ions. And obviously it's going to get harder and harder to push them through there. That's why we need the energy from the electron to do it. It's a form of active transport, and it is going against the gradient. Man, look at all those hydrogens. So a good sort of metaphor for this, and it actually works physically with what you see here, is that essentially, if you think of these little hydrogens like beach balls or something like that, we are trying to fit all the beach balls in the attic. We are shoving the beach balls in the attic. We have to do it through a little tiny trapdoor. It takes a lot of work, and we are pushing them in, even when they think they might fall back on us, but they're not falling back yet. We are shoving beach balls in the attic. Or tribbles, if you're a science nerd. That part's going to come back in a moment. All right, energy. Let's double check all of our things. So energy, we have oxidized the FADH2 and the NADH to their NAD and FAD oxidized versions. In the reduction, we made electrical energy move through these things, and we use that to create kinetic energy to push the hydrogens through. So we have an electrical energy to create kinetic energy to push the hydrogens through, and now we have a ceiling, an attic full of hydrogens, which, if you'll recall, creates another kind of energy, in this case, stored energy, positional energy, potential energy. If my attic is full of beach balls, there is a lot of potential for some energy there. And then finally, because we oxidize something, we have to reduce something. The final reduction is oxygen gas to water. So C6H12O6 becomes CO2. Oxygen gas becomes water. All right, so what are we going to do with all the beach balls in the attic? What is the deal with all of these hydrogen ions? Here's where the real fun part happens. The very last step in the electron transport chain is the ATP synthase. And here's the deal with the ATP synthase. It's the opposite direction channel from the other ones. So remember, we just pushed all of our H pluses, all of our beach balls up into the attic. What would happen if we at that point opened that ceiling attic door? What would happen to all those beach balls? They would come thump, falling down on me, which is where the tribbles come in. If you have a, compartment full of tribbles and you open it, they fall down on you like Captain Kirk. Love it. All right, so if they're going to fall, that's energy. It's the potential energy of them waiting. Then we can capture the kinetic energy of them falling. And if we can capture that kinetic energy as they fall down, we essentially smack each of them into an ADP. And that ADP is like, what? This energy is great. Picks up a phosphate group and becomes ATP. And honestly, for the ADP, that was shockingly easy. But yet that ATP is storing a great deal of energy. And this happens somewhere between 32 and 34 times. Now, it's a little hard to nail down the exact numbers. These are numbers that we give, but obviously, because of how energy works, because of the first law of thermodynamics, because of all of these things, we can't guarantee how many H's actually turn into something. We say it's usually about 3 to 1, but it's in kind of an average. We can't guarantee how many of the NADHs and FADH2s actually made it to the right place and actually broke apart. We can't guarantee any of it, but we know we create a lot. And even if our estimates are off and we're somewhere above 30, we only made 2 from glycolysis. 30 is much better. But you can see how this process entirely works. So let's back out just one more time to a quick overview of maybe not the entire thing. So as a reminder, glycolysis happens out in the cytoplasm. It takes a 6-carbon ring. It breaks it into 2 3-carbon pyruvates, which are straight chains. That pyruvate moves here into the matrix. It breaks the 3 carbons into 1 CO2 and 1 acetyl-CoA, which is 2 carbon compounds. We lose 2 CO2s. In this process, we've also lost a little ATP, gained a little ATP, and created some NADH. We then go into the citric acid cycle with our 2 carbons. Those 2 carbons get fully oxidized and become CO2s. They're gone. And we are able to capture that energy of oxidation into reduced molecules of NADH and FADH2, which then go from the citric acid cycle. The carbon just stays in the citric acid cycle except for our 2 CO2s. They go to the electron transport chain, which uses the energy of electron movement to create active transport to push those hydrogens up into the intermembrane space. Then the energy, the potential energy of the high concentration means that when they go with the gradient, so this movement is against the gradient, but when they move with the gradient down the ATP synthase, they are able to turn that movement into ATP. And because it's very similar to an osmotic process of moving with the gradient, but it's not water, it's chemicals, we call this chemiosmosis. So the entire process is cellular respiration, and specifically oxidative phosphorylation, because we use oxygen gas to make ATP, to phosphorylate ADPs by the process of chemiosmosis. Technically, you can have cellular respiration that doesn't use oxygen, but that is going to have to be for a future video. So I hope you got a sense of how your body, and actually everybody, including every tiny organism out there, every plant, everything, uses oxygen to make energy, is able to capture 6 carbons of glucose, break every one of those bonds apart, move that energy in many different ways, and eventually drop it into a whole bunch of ATPs that you can use with as little energy loss as possible. All right.